What do you do when your cameras capture a smooth, golden object attached to a rock nearly 2 miles beneath the ocean's surface? An object that looks so clean and self-contained, it almost seems out of place in one of the harshest environments on Earth. At a depth of around 3,300 meters, the ocean is a world without sunlight. There's no trace of blue, no fading glow from above, just total darkness interrupted only by the lights you bring with you. The temperature sits just above freezing. The pressure is immense, thousands of pounds per square inch, pressing in from every direction. If you were there without protection, it would be instantly fatal. Even machines have to be specially designed, with reinforced housings and carefully sealed electronics to survive. Sound is muted and strange. Movement is slow and deliberate. It's not dramatic or chaotic. It's steady, cold, and heavy. And despite the fact that this environment covers most of our planet, it remains one of the least directly explored places on Earth. What makes that even more remarkable is how connected this remote environment is to daily human life. The deep ocean acts as a massive climate regulator, absorbing and storing carbon and heat that would otherwise intensify global warming. It supports ecosystems that ultimately sustain commercial fisheries, feeding millions of people. It contains geological features, fault lines, methane seeps, hydrothermal vents that scientists study to understand Earth's processes. And stretched quietly across its floor are thick fiber optic cables carrying global internet, traffic, international banking transactions, video calls, military communications, cloud, storage. So much of it depends on infrastructure resting in darkness, thousands of meters below the surface. We rely on this environment constantly, yet we have mapped only a small portion of it in high detail and sampled even less of its life. In late August 2023, a NOAA exploration team was conducting one of many deep-sea survey missions in the Gulf of Alaska. Using a remotely operated vehicle, or ROV, they were documenting seafloor habitats as part of a broader effort to understand deep ocean ecosystems. The ROV descends slowly through the water column, its lights illuminating suspended particles like dust in a beam. Scientists aboard the research vessel monitor live video feeds, noting species sightings, geological formations, and habitat conditions. Most dives produce valuable but familiar scenes. Glass sponges rising like delicate sculptures, corals branching outward in intricate patterns, sea stars sprawled across sediment, and the occasional deep-sea fish drifting through the frame. Then the cameras moved across something different. Attached firmly to a rock was a rounded, golden dome about 4 inches across. It was smooth in a way that immediately stood out. Many deep-sea organisms have rough textures, visible polyps, branching structures, or porous surfaces. This object did not. It looked almost polished, with a subtle metallic sheen under the ROV's lights. Near its base was a small tear or opening in the surface, as though something had split or emerged from within. The edges of that opening appeared clean rather than jagged, which made it look intentional instead of damaged. Inside the ship's control room, the reaction was immediate. Scientists leaned closer to their screens. The ROV pilot adjusted position for a clearer view. Zoomed in. The object still didn't match anything obvious. Experienced marine biologists are used to strange organisms, especially in the deep sea. Even so, most species resemble known groups in some recognizable way. This one didn't provide that reassurance. Speculation began carefully. Could it be a sponge structure, some unusual coral growth, an egg case from a species not yet documented, a shed or molted biological structure? Perhaps a life stage rarely seen. Someone jokingly called it a yellow hat, a nickname that captured its shape but did nothing to solve the mystery. Beneath the light humor was genuine uncertainty. No one in the room could confidently say what it was. At that point, the mission shifted from observation to investigation. Deep-sea exploration operates under strict, practical constraints. Ship time is scheduled long in advance and costs significant resources. Weather conditions can abruptly limit operations. Equipment must perform flawlessly under extreme pressure. When something unusual appears on camera, the team has to make a decision quickly. Leave it documented but uncollected, or attempt retrieval and risk using valuable dive time. There's no guarantee of a second opportunity. After discussion, they decided to collect it. Using NOAA's ROV Deep Discoverer, launched from the research vessel Okeanos Explorer, the team carefully maneuvered a suction sampler toward the object. The robotic arm steadied the intake device against the rock. The pilot engaged suction. The golden dome trembled slightly, then detached from its anchor point. It lifted intact, suggesting it had been firmly attached but not fused permanently to the substrate. The specimen was secured and eventually brought up from a depth of roughly 3,300 meters to the surface. Once aboard, it was transferred to the ship's wet lab for initial examination. Under bright laboratory lighting, the object remained puzzling. Its outer layer was golden and leathery. Inside were layers of fibrous tissue. There was no obvious mouth, no visible skeletal framework, no clear anatomical markers, such as tentacles or segmentation. It lacked the distinctive features that typically allow biologists to quickly place an organism into a known category. It appeared biological, but minimal, almost like a structural component rather than a full organism. At this stage, many deep-sea finds remain only partially identified. The ocean floor contains countless organisms and fragments that are rarely encountered. But this specimen drew attention because it looked complete in itself. It didn't resemble a random broken piece of coral or sponge. It looked intentional, like a structure built for a purpose. The sample was later sent to specialists connected with NOAA Fisheries National Systematics Laboratory at the Smithsonian's National Museum of Natural History. Institutions like this are not simply museums in the public sense. They are research centers, housing vast collections of preserved organisms used for scientific comparison. Identification often depends on matching unknown specimens against known references stored in these collections. The first analytical step focused on morphology, the study of form and structure. Scientists prepared thin tissue sections and examined them under microscopes. That's where they found a critical clue: cnidocytes. Cnidocytes are specialized stinging cells found exclusively in cnidarians, the group that includes jellyfish, sea anemones, and corals. These cells contain microscopic capsules capable of firing thread-like structures used for prey capture or defense. Their presence immediately narrowed the possibilities. Whatever the golden object was, it belonged within cnidaria. Further microscopic examination revealed spirocysts, a specific type of cnidocyte characteristic of certain members of Hexacoralia, a subgroup that includes many sea anemones and stony corals. Spirocysts are adhesive rather than penetrating, helping organisms attach to surfaces or secure prey. Finding these cells significantly refined the classification. The object was very likely associated with an anemone-like lineage. However, microscopic features alone could not determine the exact species. The deep sea contains many poorly studied cnidarians, and subtle structural similarities can exist across different taxa. The next step was genetic analysis. Researchers first attempted DNA barcoding, which targets short, standardized gene regions commonly used for species identification. This method is efficient and often effective when reference sequences exist in databases. In this case, the results were inconclusive. Deep-sea specimens can contain mixed DNA from microbes or environmental sources. The golden object was not a pristine, intact animal, but appeared to be a remnant or structural component, making clean sequencing more challenging. To obtain clearer results, scientists turned to whole-genome sequencing. Instead of examining short genetic fragments, they analyzed much larger portions of the specimen's DNA. This approach produces a far more comprehensive dataset and allows for stronger comparisons. The genomic data showed strong alignment with Relicanthus daphneae, a giant deep-sea anemone. Relicanthus daphneae is a large anemone found on the ocean floor, often associated with hydrothermal vent regions. It was reportedly discovered in the 1970s, but not formally described until 2006. The species can grow large, with tentacles reported to reach 6 to 7 feet in length. These tentacles extend upward into the water column, capturing passing prey in the darkness. The key realization was that the golden dome was not the visible body of the anemone. Instead, it appears to be part of its anchoring base. Tissue, combined with a durable, cuticle-like outer layer that helps the animal attach securely to rock. The anemone's main body sits above this base, which is normally hidden from view. Because of that, researchers rarely observe the underside directly in its natural setting. When the animal is present, the base is concealed beneath its column and tentacles. What the ROV discovered was essentially a leftover foundation. The object was first observed on August 30th, 2023. It took until April 2026 for researchers to publicly close in on a confident identification. That extended timeline reflects careful laboratory work, microscopic analysis, genetic sequencing, database comparisons, and evaluation against museum specimens. Science, particularly taxonomy, often requires patience and multiple lines of evidence before drawing firm conclusions. The interpretation was strengthened by comparison to similar specimens collected previously, including material from a 2021 expedition aboard the research vessel Falkor. That earlier sample displayed matching cellular structures. When independent specimens share consistent microscopic features and genetic signatures, confidence increases significantly. One unusual object might be misinterpreted. Multiple consistent examples establish a pattern. Even with identification largely resolved, questions remain. Did the anemone die at that location, leaving its anchoring base intact, as the rest of its body decomposed or detached? Or is it capable of relocating, abandoning older attachment layers as it moves to new substrates? The golden object's identity is mostly understood, but aspects of the species' behavior in the deep sea remain open for further research. This entire process demonstrates how deep-sea science unfolds in reality. Discovery begins with observation and confusion. Confusion leads to collection. Collection leads to microscopic and genetic analysis. Data is compared against reference libraries built over decades. Only after multiple independent lines of evidence align do researchers feel confident presenting an answer. In the end, the mysterious golden object, 2 miles underwater, was not a new species, not an alien egg, and not an unknown life form. It was a structural foundation, an anchoring base from a giant deep-sea anemone. And if something as small as a 4-inch attachment structure can take more than 2 years to confidently identify, that underscores how much remains to be learned. The deep ocean is not empty or simple. It is layered with detail, and we are still developing the tools and patience required to understand what we're seeing.